Good afternoon, everyone, and welcome to our monthly Regenerous webinar. Today we're being joined by Emma Beswick from Lifecode Gx, and she's going to be discussing the very fascinating subject of genetics, specifically the what, why, and how of DNA testing. We're also excited to announce the new and exclusive partnership we're going to be having between Regenerous Laboratories and Lifecode Gx. We're sure there's going to be a great partnership. The detailed genetic panels offered by Lifecode Gx are undoubtedly going to support the extensive range of complementary tests we already offer. I'm sure that many of you already know Emma, but for those of you who don't, she's a very experienced nutritional therapist and a member of BANT, the IFM, and the International Society of Nutrigenetics and Nutrigenomics. Glad I got that right. Emma is the founder and director of Lifecode Gx, which provides DNA testing, analysis, and consulting for professional use. She's also a strong advocate of an integrated approach to optimizing health. So this involves looking at the whole person, including genetics, family, health history, symptoms, nutrition, and of course, lifestyle to empower individuals to improve their physical and mental health. I'm sure you'll all join me in extending a very warm welcome to Emma. Hi, Emma.
Hello.
Now, I know you've got an amazing presentation for us, but I thought we could just start by hearing a little bit about how you started your journey into the wonderful world of genetics, and also maybe how you identified a gap in the market for what you do with Lifecode Gx.
Sure, thank you, Charlotte, for that very wonderful introduction. So I first was introduced to nutrigenomics when I was studying at CNN, and we were very fortunate to have a lecture on that subject, and it was quite advanced for colleges to be teaching that at the time. And straight away I was interested, it just grabbed my attention. I went and did a test for myself. At the time I used a company called 23andMe. And I then used that information to try and make some sense of what was going on with me and with my family. And I had some limited success with it, but what I found was that it took an awful lot of time to really get anywhere. And what I was given was some fairly trivial statistics about, can I taste bitterness? Is my earwax wet or dry? What eye colour I have, which I did already know.
Very useful.
No. And I obviously, as a trainee nutritional therapist, was really most interested in the health aspects. And I found that it was quite limiting and very time consuming. And so I saw a need for a professional product in that space that was tailored to the way we think and the way we practice as functional medicine or nutritional practitioners and professionals. So that's what I hope we have created, and I do believe we have created with our Lifecode Gx tests. And the other thing about those other products is that it was quite an inconvenience to do a test to get some data and then another exercise to interrogate that data and interpret it. Whereas what we're able to do is offer a one-stop shop where you basically buy a test just like you would buy any other functional test. You buy it from a trusted provider, you get the results, you get support in interpreting those results. And so it's all a much more secure, streamlined, reliable and trusted process. So I think that's where we are now. And as Charlotte said, we are really delighted to be working together to be able to work in an integrated way and practice what we preach and get the best of those products from one place through Regenerous, and offer that opportunity to do that at your convenience. So let's get started. So you know a little bit about us already. Lifecode Gx has been around for about 2 years now, so we're still quite young, but we've made some pretty big inroads into the nutritional therapy market, because we do stand out in terms of what we offer. So hopefully that will become apparent as we go through this. So I'm going to cover the what, why and how of genetic testing, and then to give you some insights into how that can be used in practice. Firstly, looking at individual genes and the benefits of that, but also the limitations. And then most importantly, the connectedness of gene to gene and looking at that more complex web of information, the genes relating to the genes, but also to the cofactors and also to the wider environment that a person lives in. We're going to look at some specific test panels as examples. We're not going to be able to cover them all, so it's just to give you an insight at this point into some of the things that are available. And then we'll talk a little bit about practitioner support and education and where you can find out more. Okay, so to start off with, why would someone do a genetic test, and why would you recommend a test to your client? So some of the answers to that question are going to evolve as we go through the presentation. But first of all, curiosity is probably the main reason that the person on the street embarks on a test. So the big growth in direct-to-consumer tests is really because people are curious about their ancestry, possibly more than their health. They want to know what percentage Spanish or Russian or Asian they might be, or whether they're Neanderthal or not. So those are really direct-to-consumer test focus. The other points are much more areas that we would look at, or reasons that we would state as nutritional therapists or health practitioners. So really to get to the root cause of an existing health issue that maybe has been quite elusive, to be proactive in terms of prevention of something happening, particularly if there is a family history, for example, or maybe to optimise health. If someone really wants to be top of their game and perform at an executive level or in an athlete kind of way, or even just for each individual to feel that they're optimising their own health and being able to live their life to its max, really. So the 2, 3 and 4 are the reasons that we as health professionals would look at this. But you may well find that your clients come to you having already done a test or part of a test or heard about it, and they're looking at it from also the first perspective. Okay, I'm going to just give you a very brief intro into what genetic testing is before we look at the tests themselves. So firstly, the human genome. The little diagram on the right there is illustrative of the different components of the genome. So at the top level, the genetic code is held on chromosomes. So we have 23 pairs of chromosomes. That's been compared to a book. The chromosomes contain genes, and those could be thought of as being sentences. And those genes are constructed from codons, which are made up of 3 bases, so 3 letters, which code for an amino acid. And then the amino acids basically group together, chain together to make proteins. So at a high level, the genes code for proteins, that's what they do, and then the proteins have various functions. And we have 30,000 genes approximately. Those are the known genes in the human genome, and 3 billion base pairs. So those would be A, T, Cs and Gs. So a genetic test actually aims to identify the variants in someone's code, and it's comparing each individual to a actual mystery, non-possible perfect person That's what the tests do. They look for variants, but the perfect person doesn't exist. It's just useful as a reference point to see how someone's genome compares to what would be the perfect or most common combination of codes. So a genetic variant is a change, or it can be an insertion or a deletion of some code. So that can either be a single letter, it can be a group of letters, it can be a whole gene, or it can be a whole chromosome that is either missing or duplicated. And sometimes those things do make a big difference, and sometimes they don't make any difference at all. Particularly if it's just a letter, you can have letter changes that don't have any effect. You're probably familiar with the term SNP, which is short for single nucleotide polymorphism. And this is what we tend to look for at Lifecode Gx. We're tending to look for these because they make a difference, but they don't make a difference that can't be adapted to or adjusted for by nutrition and lifestyle. So we're looking for things that make a difference that can be actioned. So what you see here at the bottom, we have a wild type, which is made up of a T, a G and a G, and that codes for the amino acid tryptophan. However, if that middle G is changed to an A and someone has an A in place of that at that location of their genome, instead of coding for tryptophan, it codes for stop. So instead of all the amino acids chaining together to make the gene and carrying on after tryptophan, it just says stop. So that gene would be curtailed and shortened at that point, or the protein would. So you can see in that case how if the protein is a different shape, then it would probably affect its function, and it might affect it to the extent that it has no function whatsoever, so that gene doesn't work at all. So that's what a SNP can do. Alternatively, a SNP might make no difference. It might actually not even change the amino acid that is coded for, because you can have amino acids that are coded for different combinations of bases. Okay, so all tests need a sample, and that can be blood, saliva, or cheek cells, or hair, or lots of other bodily substances, but those are the most common. We use cheek cells because it's easy and painless and can be done at home. And I think one of the key things to mention here is when you're looking at companies and deciding who to test with, be aware of how that sampling is stored, what is done with it, how secure is it, how private is it, how is it kept, and how long is it kept for. So for us, our lab is in the UK. Our samples are kept for 3 months, just in case there's a query about result, that it's kept for 3 months. And also we can retest using the same sample. And then after 3 months, that sample is destroyed. So there is no way anyone could get hold of it. And the other thing is we never send a client ID or name to the lab. They don't know anything about the client. So there is no risk of that sample being associated with an individual. So that's sampling. And then the next stage is sequencing and genotyping. So effectively they almost produce the same thing. Sequencing is focused and it's much more extensive. So sequencing involves looking at every single piece of code within either a targeted test that looks at a specific gene. So say you were looking at BRCA genes for breast cancer risk, sequencing would look at every single piece of code on that gene, whereas genotyping would just look at certain specified codes, specified locations to see whether they were different. And whole genome sequencing is something that's still very expensive. It's about $1,000 to do, and it churns out the 3 billion bases that we talked about. So it's very unwieldy in terms of data. It's still not the norm. Whereas genotyping is what most test labs do. It's more manageable, it's more focused to test for specific differences. Okay, and this is what comes out when you do a test. This is the raw data. So it's important to understand what this is, because the reference sequence on the left there is the actual key to what you're looking at. That is the one thing, it's like the identifier in terms of knowing that you're talking about the same piece of code when you look things up in research or see it on a report. And I would say if a report doesn't show that information, I'd be slightly concerned about really knowing what it is they're testing for. So we're very transparent about that information. It's included in the report. The next information on there is what chromosome we're looking at, and then the other digits of the location of the chromosome. And then finally, the genotype are the two letters that are present for that person at that particular location. And that is what we analyse, and that's the real key information. So it's the reference sequence and the genotype that are most important to us. We take the raw data, we compare it to our curated research database, and then we put it through our systems, our apps, and generate a report, which is an interpretation of that raw data. It's fully referenced, it's very transparent, and you're going to see some examples of that. And then very importantly is the application, which is what we do as therapists. So that's basically what, so you get the report, the client's got their report in their hand, or you've got their report. It's what you do with it that is the most important thing. So first of all, as we introduce the reports, I'm just going to explain some different ways of representing those genotype results, because they are different depending on which test company or interpretation company you use. So you will usually see an allele result, which is the two letters. You might then see the genotype described in a number of different ways. So it could be described in a narrative way, like wild type. It could be described using pluses and minuses. Minus usually means no change, but I still find that quite confusing to really be confident which way around it is. And then often colour coding is used, with green most often used for the wild type, because the wild type usually means that's the most common and it's usually beneficial. But that's not always the case. And we actually, in our test results, use colour coding to represent the meaning of the result rather than just whether it happens to be different or not. So that you can use that colour coding to know intuitively that's where to focus on to help your client the most. Okay, so this is typically how we would represent a detailed result for a client. So you can see on the left we provide the gene name. There's the reference sequence ID, which you saw in the raw data example, the genotype, the colour coding. In this case, it is a homozygous, it is a variant, and the variant is associated with a negative meaning. And in this case, we're looking at a gene that is responsible for recycling vitamin K. And this result means that this individual is less efficient at recycling vitamin K. So there is more risk of them being insufficient or deficient in vitamin K. But I think the key thing here is transparency. Again, you want to know exactly what's being tested. And then you can also look at it for yourself to make sure that that is valid. But we provide the links to the references anyway, so you can just click on those links and see why we are saying what we are saying. At the same time, you don't need to do that because you've got the key information in front of you. Okay, so A little bit about context. So one thing that I cannot emphasize enough is the importance of context. So just because you get a result that says someone has a predisposition to be deficient in vitamin K because they can't recycle it efficiently, doesn't mean they are deficient in vitamin K. They might already be doing all the dietary things to address that and to compensate for that genetic variant. So I really, really, really caution against taking a test result or even reading or trusting a company that says this test result means you need to take more vitamin D. For example, it's irresponsible to make those kind of statements just off the back of a gene test, and especially a single gene. I think all the genes that we look at are, because they're variable and actionable by diet and lifestyle, it's vital to put it in context. So you've got the predisposition, and here we have the functional medicine tree, which some of you have probably seen before. And genetic predisposition is right near the roots there, but the genetic predisposition needs to be considered alongside epigenetics, so the environment and the interaction of the environment with those genetics. So it's important to look at what's going on with a person just exactly as you would in practice with someone who wasn't or didn't have a genetic test result. But you have the benefit of that extra information. So that's how it should be seen, as extra information, not just information in isolation. Okay, so to really emphasise the point of that, I think genetic testing is a perfect fit for functional medicine and nutritional therapy, because we've all been trained to look at it in that context and to look at the whole person and to look at those different dimensions. So the family history, the person's full health history, the symptoms they might be experiencing, and also other functional test results. So that's incredibly vital. Okay, so a little lesson on terminology. So nutrigenetics and nutrigenomics are often used interchangeably, but they are slightly different. So nutrigenetics is about the effect of genetic variation on dietary response, and I'm going to show you some examples of that in a minute. Nutrigenomics is more about the role of nutrients in impacting gene expression, but we generally do work with both. A lot of the more simple direct-to-consumer tests focus on nutrigenetics, and it's quite simplistic. I think it really, really comes into its own when you're looking at a much more dynamic scenario, which is the bigger picture, and hopefully you'll see that in a second. Okay, so the nutrigenetics, here are some examples of how gene variation can affect response to environmental or dietary factors. So if you've got a certain form of lactose gene, then that can affect your ability to digest lactose. HLA affects response to gluten, DYP1A2, caffeine, vitamin K we've talked about, and so on and so on. You can see it's not exclusive to food. So one of my favourite genes, I think just because I love the name, is the clock gene, which affects people's response to light and their circadian response to light. And then we've got an adrenaline receptor that impacts someone's sensitivity to adrenaline, and really that fight or flight response, and how keenly that might be felt by different individuals. And that's certainly something that is of interest to me personally. I do have an exceptionally sensitive adrenaline receptor. And then there's also IFNG at the bottom, which is our response to viral or bacterial infections. So it's very varied. Okay, so an example to just drill into is lactose. I think this is a nice ease into how we look at things. So in this case, the gene is responsible for acting as an enzyme. So it's converting lactose to galactose and glucose, and it's enabling a person to digest lactose, to break it down. So if there is no variance by default, after someone has been weaned, that gene actually switches off. And this is the default position that humans started off with. And they would become lactose intolerant after they'd stopped breastfeeding. So the variant on the gene in this case actually allows the lactose gene to continue to be expressed. And so that means that people can continue to digest lactose. And that actually evolved over hundreds or thousands of years as people started to farm animals. So you find that Caucasians have more often are able to digest lactose for this reason, because they have the variant, but only about 2% of Asian people can. And the way that would be reported could be like this. So in this case, the wild or the default is actually color coded as red, because that's seen as negative in respect of someone's ability to digest lactose. Whereas the active version of the gene is associated with the variant, and so that's color coded green. And as an adult with this genotype, you would expect adults to be able to digest lactose. Doesn't mean they might not have other problems digesting milk or eating dairy, but certainly from the lactose perspective, that shouldn't be a problem. So that's a very simple example. Before we look at nutrigenomics, so this is how the nutrients can affect gene expression. And we are going to really go into some examples for this, but some examples there at a top level are the effect of methylfolate or methyl B12, which you might have heard of. So these are activated forms of those nutrients that are responsible for methylating genes or methylating other substances. Often methylating a gene actually suppresses its activity, but that's a good thing, or can be a good thing, because if you think our DNA is in every cell of the body, we don't want a neurotransmitter gene working in the liver necessarily. So methylation is one of the mechanisms for making sure the genes that don't need to work and shouldn't work in a particular place are switched off, but the right ones are switched on, as well as facilitating lots of other reactions. Vitamin D is a cofactor for serotonin synthesis. Magnesium is a cofactor for the MAT1A gene, which is the last step in making SAMe, which is the master methyl donor, which we'll come to. And then substances like curcumin actually inhibit activity of the MAO-A and MAO-B genes, which are neurotransmitter metabolizers. So by inhibiting that activity, curcumin can act as a natural neurotransmitter supporter by slowing down the metabolism of those neurotransmitters via its action on that gene, and so on and so on. Brassicas are absolutely huge in terms of their impacts on genes and controlling them, supporting them, or dampening them down as appropriate. So I'm going to show you an example of a nutrigenomic situation. So vitamin A, vitamin A conversion from beta carotene to retinol. So the activation of the vitamin A is controlled by a gene called BCO1. And BCO1 SNPs or variants can actually reduce the rate of that conversion by up to 57%. And this is just 2 SNPs on that gene can have that effect. So those 2 SNPs basically can have the effect of slowing that gene activity down. Now, the reason this is in the nutrigenomics section as well as the nutrigenetics section is, from a nutrigenetics perspective, the gene is affecting the response to vitamin A, or the ability to utilise it. But then, to add to that, if you look at what retinol is used for, so people might think, oh, so that's really simple. We're just looking at one gene and a couple of SNPs on the nutrient core report, and it's telling us about vitamin A. But look at all the things that vitamin A is needed for. So it's needed for eyesight, we know that. It also affects metabolism. It also affects steroid hormones in terms of hormone transport into the cell. Absolutely massive in thyroid, absolutely massive in terms of immunity. So you look at one thing that looks quite simple, and actually one simple thing, one simple SNP or 2 simple SNPs could have a very, very big knock-on impact in terms of the effects on other genes and other functions. So the retinoic acid actually affects the expression of those receptors that are shown there, the RAR and the RXR, and their response in context to those other substances, thyroid hormones, vitamin D, glucose, other steroid hormones, etc., etc. So that's why it's really important that someone as a practitioner has all this knowledge, whereas someone on the street might not think that it really mattered that much. Okay. Histamine. So we have an entire report on histamine, and I'm going to use this as an example of quite a simple report to show you some features of how we analyse genetics and how it makes it easier for the practitioner to use. So just a little bit of background about histamine. We need histamine. We don't want it to be too low. It has lots of really, really important functions and reasons to be there. But most often people are likely to complain or experience symptoms or problems associated with high histamine rather than low histamine. And here are some of the symptoms that might be experienced. This is a summary of the histamine degradation pathway, and it's a typical of a pathway analysis that we would use. So at the top is the synthesis part. We're making histamine. That's generally not genetically where the problem is. The problem is someone's ability or otherwise to break down the histamine and to remove it at a good enough rate so that it doesn't overwhelm the system and create some of those symptoms that we just saw. So what you can see here is histamine can be broken down in 2 different pathways. On the right hand side, we've got the DAO route. So the gene there is in bold letters, DAO, and that is mostly responsible for degrading histamine in the gut. And the key things here are this person actually has got a result indicated by those little green circles that is a really positive result, and it means that gene should be functioning normally. There's no genetic reason why that wouldn't be. But you can see alongside that, there are some purple blobs and some orange blobs. and the purple are cofactors. So in order for that enzyme to work optimally, it needs sufficient supply of vitamin C, B6 and copper. And then in addition to that, there are some orange blobs which are inhibitors. And if there are too many or too high volume of those inhibitors, then that's going to slow down that pathway and prevent it working optimally. So it's about taking all those things into account to optimize that person's pathway. So they could still have a green result genetically and not be exhibiting symptoms of gut histamine overload. But if they've got red results on that, it's ever so much more likely. And then on the left-hand side is HNMT, and this is much more involved in histamine higher up the system. So histamine is actually a neurotransmitter as well as a kind of immune responder, and it's involved in brain histamine. It's also involved in airway histamine. So the kind of symptoms that someone might experience would be shortness of breath, asthma type symptoms, runny eyes and nose, etc. And again, the coloured dots are indicative of genetic SNPs, and in this case, again, it's green. I haven't chosen the best example, but the key thing here is that the cofactor is really vital. So the cofactor there is SAMe, and SAMe is the master methyl donor. So for this pathway to work very well, it's got to have sufficient levels of SAMe. And to have sufficient levels of SAMe, you need to be methylating sufficiently, which we'll come to next. Quite often, histamine and methylation are opposed in terms of if someone has very high histamine and symptoms of high histamine, it's indicative that their methylation is suboptimal. And there's a bit of chicken and egg there. So one is that there's not enough SAMe to be mopping up the histamine and helping this pathway. But conversely, the methyl groups, the SAMe, may well be being depleted by excess histamine. So the methylation as a whole suffers, and that has a whole range of effects on lots and lots of systems. So high histamine is associated with low methylation. So that's a really, really worth remembering thing. Another little hint is that the gene HNMT, an MT gene is a methyltransferase. So without having to know every single gene that needs a methyl group, if you see MT as part of the name, that means that it needs SAMe as its cofactor. So that's just a handy little tip there. And then you'll see lower down the pathway, there's the MAOB gene, which is responsible for a lot of neurotransmitter metabolism. And look how many cofactors that MAOB gene needs. So zinc, magnesium, B3 and B2. So if that's not working, and the yellow dot here next to MAOB indicates that it would be slower and less efficient, then that's not going to be breaking down histamine. So it's still going to be there, going to back up, and the whole histamine picture could well be affected just by that one SNP. Okay. So I'm going to give you a very rapid insight, or for those of you who've been to our seminars and know a bit about methylation, a little bit of a reminder about methylation, because it is so important, and genetics can provide such a massive insight into methylation. So quite simply, what is methylation? It's the addition of a methyl group, so a little CH3 group there, compound, to a substance which methylates the substance. So we're going to look at homocysteine in a minute. Homocysteine gets methylated and it becomes methylene. So Methionine is just a methylated form of homocysteine. Methylfolate is a methylated form of folate, so it's folate with the methyl group added. Trimethylglycine is methylated glycine, it's also called betaine. And then samine is a methylated form of methionine. But all these things here, the methylfolate, the betaine and the samine, they've got their methyl group, and that means they can then donate it and help another reaction to take place by passing that methyl group on. So that's what methylation is. It's the addition, the passing on, the donation of a methyl group to a substance to make it active or do something else, quite simply. So this is a high-level overview of the first two cycles of methylation. They are the core cycles for making samine, and so hence I always think of these as being at the heart of methylation. They're the synthesis part. So on the left-hand side, we have the folate cycle, and we take folate into the body, and then it goes through lots and lots of different conversions to become trihydrofolate. Different forms of trihydrofolate, and there are lots of genes involved in that we don't have time to go into here, but I mention it because it's important to take those into account. And then the 5,10-methylene trihydrofolate, THF, is then converted to 5-MTHF, which is another name for methylfolate, and the MTHFR gene is the gene that is needed to do that, or the protein that is needed to do that. So people quite often get very excited about MTHFR SNPs and all very worried. So especially if people have done a gene test on their own without the support of a practitioner, they might get an MTHFR result and think that something terrible is going to happen, or that that's the reason for lots of bad things already happening to them. And that, unfortunately, is a risk of people doing genetic testing from having a little bit of knowledge, and that potentially being a dangerous thing. So it's really, really important, firstly, to take into consideration the other genes. Secondly, they're not shown here, but there are cofactors around MTHFR, which are B2 and B3. And so they will also affect the action or rate of the activity of that gene, as well as the amount of folate that you're giving it to work with in the first place. So MTHFR is important, but it's not the be-all and end-all. Once you've made your methylfolate, or if you've supplemented methylfolate, that methylfolate then goes up and is firstly used to methylate B12. So you see there's a little mini cycle with a purple B12 and a purple methyl B12. So the first job of the methylfolate is it methylates the B12, and then that methylated B12 is used to methylate the homocysteine and make it into methionine, and the MTR gene facilitates that makes it happen. So how many people out on the street really talk about MTR? They don't, but the MTR gene is just as important as the MTHFR gene in terms of how is that working, how efficient is it. You might have lots and lots of methylfolate, but the MTR gene can affect how efficient you are at using that methylfolate, as can the amount of methylated B12. So if you don't have sufficient methylated B12, then the methylfolate isn't used properly. If you've not got B12, the methylfolate has nothing to work on, so then you can't use the methylfolate, you can't use the B12, and you can't convert the homocysteine, and as well as if you had all those things in the right proportion to each other. So the important thing is to not just focus on one thing, not one gene, look at all the genes in combination, look at all the substrates and cofactors in combination, and make sure they're in balance and in the right proportion in order for this cycle to work in terms of that homocysteine being recycled to methionine. And that's just the start point. And I appreciate that I've whizzed over this and it's not intended to be an in-depth tutorial on this, because we spend a whole day on a seminar teaching people about methylation, and this is probably, these two cycles probably take at least an hour, maybe longer, to go through properly and explain all the genes and cofactors and how to balance them and what the different forms of folate and B12, etc. are. So once you've made your methionine, the methionine then needs to be converted to SAMe, and that needs the help of the MAT1A gene on the right, top right there, and the cofactor for that is magnesium. So again, someone might have SNPs on MAT1A, which mean magnesium, magnesium, magnesium is really, really, really important to support that pathway the best you can, and also to make sure they've got good amounts of methionine in order to make the best of that situation. But as I said at the beginning, none of these genes are deterministic in that they're not going to tell you that you can't do these things, that you can't methylate. We can all methylate and it can all make SAMe. We just might need different levels of support and different types of support to make that happen to the optimal we can. So this is just to show you the folate cycle in detail. I'm not going to go into all that detail, but this is what I meant when I said there are lots more genes involved, lots more cofactors. You can see the purple blobs, there are B3, B2, B6, all really, really important to work alongside folate to optimize the methylfolate synthesis, but also the folate cycle itself, which is really, really important for DNA synthesis and repair, really important for healing, really important for prevention of cancer, really important for fertility and pregnancy. So that's a snapshot of the folate cycle in all its glory and all its genes and nutrients that are needed for that. And similarly with the methionine cycle, this is a zoomed in view. This is what you get from a test result in terms of the detail and the other genes that are so important as well. And then behind that, there is also a personalised explanation of what all those mean. And of course, we don't want you as a practitioner to feel overwhelmed by this. So that's why we do provide support and we do provide training. The point I made about the MT genes earlier, the ones that are methyltransferase and have MT in their name, and therefore need SAMe or another methyl group as a cofactor. This is an example of some of those MT genes. It is by no means exhaustive, but it's here to show the many reasons why methylation matters. So if methylation isn't working optimally, if it's sluggish and there isn't much SAMe around, then these are all the genes and all the functions that can be impacted by that. So COMT, very important for breaking down estrogens and neurotransmitters like dopamine and noradrenaline and adrenaline. So if someone's experiencing a lot of stress or symptoms of estrogen dominance, then you want to be looking at COMT. You want to understand that person's genotype for COMT and how efficient that gene is likely to be in the first place, and then also examine methylation in respect of the supply of the SAMe and the cofactors to support COMT. And the same with all these other genes. We talked about HNMT in the histamine intro, but you can see there it can affect sleep, melatonin, choline, detoxification, adrenaline, metabolism, lots and lots and lots of things. So that's why methylation is such a core thing to understand. We know that genetics can affect methylation very significantly, but we also know that we can adjust for that and we can balance that out if we know exactly where and how that methylation synthesis, that cycle is in need of help. Okay, so that's methylation in 5 minutes, and I've got 2 more examples to show you. One is estrogen balance, and we all know that there are quite a lot of symptoms of hormone imbalance. These are just some of them. And you might well be used to using a test like the Dutch test to look at hormone levels. The difference between a genetic test and a functional test, like a Dutch test, is a genetic test will tell you your predispositions, and your genetics, the ones that we're reporting, never change. So you have them when you're born. Your genetic code does not change. So once you've got that test and you understand your pathways and your predispositions, you've got that information, that's it. The things that might change are our knowledge about what those variants mean is improving all the time, but the actual code is the code. That is with you forever, and it's showing you what will always be maybe a vulnerability or a slow or a fast predisposition or for a particular pathway. Whereas a functional test usually shows you something that is temporary or maybe temporary. So it's giving you a snapshot at a point in time of what your levels of a certain biomarker are. Now, it gets very, very, very powerful when you put the two together and you can see that someone maybe has a functional test that says they have a high amount of, let's just take an example here. On the estrogen diagram, on the right-hand side, near the CYP1B1 SNP, we have a 4OHE12, so second up from the bottom on the right-hand side, 4-hydroxy estrogen. So this type of estrogen, this form of estrogen, is the one that's associated with damage and cancer risk and oxidative stress. So in general, we don't want that pathway to be working any quicker than normal. And especially if someone has high estrogen anyway. Now, you might do a Dutch test and that test might tell you that you've got a high level of 4-hydroxy estrogen. If you also have a genetic test, that genetic test might back up and it might actually correspond very closely to what the functional test is telling you. And it might tell you that the CYP1B1 SNP is actually fast. That's what this genetic test might tell you. And the Dutch test might tell you the same, in which case it's like, okay, I've got a fast acting gene pathway at the moment that's causing my 4-hydroxy estrogen to be high, and it's genetic. So that's something that you know is innate, it's a predisposition. However, we wouldn't be telling you about it if you couldn't do something about it. So we're telling you that you can do some things to reduce that activity and to reduce your levels of 4-hydroxy estrogen. Like grapefruit juice slows down that CYP1B1 pathway so it doesn't work as quickly, for example. Now, if you know that there's a genetic aspect to that, then that's much more of a long-term view. that you should be thinking about things that you can integrate into your lifestyle or your client's lifestyle as habits that are for the long term. Whereas if it's something that isn't consistent with the genetics, then it should be something that you can fix quite easily. And it might be temporary because someone's actually under a lot more stress, for example, than normal, and it's pushing that pathway. There's something else going on that is promoting that, but it shouldn't be something that is a long-term thing. So that's just a little lesson into the two things together and what the difference and what the similarities are between genetic testing and functional testing. And from a genetic perspective, this is another pathway. It's a drilling, but what it's showing you is at the top, we've got some things involved in synthesis, so the CYP19A1 and 17A1 genes. In this case, the red means they work more quickly, likely to be upregulated. So for a start, this person is more predisposed to making higher levels of estrogen. The middle group is what forms of estrogen are more likely to be dominant. In this case, it's fairly balanced, although the 2-hydroxy, the middle one, is a little bit slow because of those CYP1A1 SNPs. And then the COMT, which I mentioned earlier, is the step in methylating that estrogen. So it's converting the hydroxy form into the methyl form, and it's deactivating it. So that COMT result means slower. And we know that COMT needs SAMe, so we need methylation to be working well to almost compensate for that COMT SNP and to give it all the support you can in terms of helping it out. And then at the bottom there is the final stage in metabolizing those substances. The GSTs on the right are glutathione transferase genes. They're important for antioxidant action in terms of they use glutathione to get rid of oxidative stress products, free radicals, carcinogenic substances, for example. And the red result there means that these don't work particularly well and they need a lot of help or would benefit from help by reducing toxic loads, but also by supporting with a cofactor like glutathione. And the information on the left here includes some of the nutrients that you might consider to actually support those results. So this has been recorded, hopefully, so you'll be able to look at this, and if you're interested in this, you can pause on it and look at it in more detail. So I'm going to give you a very quick insight into nervous system, and this is the final insight. So we're quite unique in offering a genetic test that looks at nervous system. We look at neurotransmitters specifically, and these are some of the symptoms of neurotransmitter imbalance. So most people at some point in time experience some of these things, and that might well be okay if it's short term and depending on the extent of it. But for some people, it's outbalanced by a long way, and it can be outbalanced for a long time. And some aspects of that are likely to be genetic, and some aspects are likely to be environmental. And we can put the two together, again, by looking at the genetics and the epigenetics to see how that might be affecting someone. So this is just what we cover. So the main neurotransmitters and also thyroid hormones. So we look at the genes that basically activate thyroid hormone from T4 to T3. And this is a serotonin pathway. So hopefully you're familiar with the pathway idea by now. This is basically showing that the synthesis is slightly slower because we've got all those amber SNPs from tryptophan to 5-HTP that are slowing down that first step from tryptophan to 5-HTP conversion that is needed to make serotonin. But also, this person on the right-hand side at the bottom has got a slower MAO gene. That's red with the two arrows down. And that might well balance things out. And the inhibitors shown there, curcumin and quercetin, slow down the MOA further, whereas the cofactors, the B2, the magnesium and the zinc, speed it up. So it depends what you're wanting to achieve with this person and what symptoms they're experiencing in terms of high, low. And this can help you work out how to adjust for that. And then finally at the bottom there, the ASMT gene, which needs SAMe, is needed to actually synthesize melatonin. So people have got difficulty making melatonin. Methylation again could well be a factor in that, alongside the specific gene that does that activity, the ASMT. Okay, and then this is a page from a report which provides information about follow-up and testing and nutrition and lifestyle, and all the reports include that context and that advice. Okay, so that's the insights, and very, very quickly, I didn't go into everything because there's far too much, but these are the reports that we currently offer. These are the tests that we offer. So you can basically get one test. You can then have the option of adding on a second test when you get the results for the first one, so you can see what comes out of the first one, have a discussion with us and with Genus about making sure you understand that, and then you can decide whether you need or would like more insight either to another genetic test or another functional test. And then this is just a little reminder of some of the key points to look at when you're looking at test companies. So data security, client identity, protection and respect for that. And then also what standards of accuracy does the lab have? What sort of testing and sequencing do they use? How do they validate that? Is it checked, etc.? Then what is the interpretation like, if anything, or do you just get a big grid with a lot of colours and pluses and minuses? We really have put a lot of effort into the interpretation and providing the context and the advice, and references. So you can see there at the bottom just a little screenshot of references. So all our reports have the references in there as hyperlinks, so you can see, you can click through to the research papers to find out more about those specific genes and SNPs. And then finally, practitioner support, which for a new science, which I think nutrigenomics is still quite new, it's vital to have training and support. So we do provide one-to-one support, and we also provide practitioner training. So here are some of the training events that we have lined up for this autumn, and you can find out more on our website. They're all CPD approved. They're all a day, Fridays or Saturdays. The London ones are at the Royal Society of Medicine, and we've got a Manchester one also in November. And we've got a few tickets left for Nutrigenomics 101 and about one ticket left for the Brain Health one at the moment. So that's that. And then finally, this is how to contact us, and this is how to contact Regenerus, just as a little reminder for you there. And I think that's it. So I'm going to stop sharing my screen, and I think we're going to go through to Q&A, just to see what questions have been put there, and I can try and answer them. So, can you tell symptomatically if your CYP1A2 is up or down regulated? So CYP1A2, I'll just remind myself which one that is. So I think the 1A2 is the gene that metabolizes caffeine, off the top of my head. That basically, if it's fast, it means that you should be able to remove caffeine from your system more quickly. If it's slow, it means that the caffeine will probably stick around for longer, and you are more likely to suffer from sleep disruption if you have caffeine later in the day. So that's one way of telling. With caffeine, it's quite interesting because there is a lot of focus on the CYP1A2, but it's not just about CYP1A2. There's also a gene called ADORA2A, which affects your sensitivity to caffeine. So if two people sit and have the same dose of caffeine, then someone with a ADORA2 SNP will more likely feel much more caffeinated than the other person, and that's basically often a greater impact on someone's caffeine response than the 1A2. Okay, so I'm just looking quickly at the questions. Lifecode Gx training, hopefully I've answered that. So we have a number of seminars. You can see those on the website as well at lifecodegx.com events. So that's always there, it's always updated. For the test costs, it depends. So you can register as a practitioner with Regenerus, and you can register as a practitioner with Lifecode. And you basically, the test generally retail, the retail price that the clients see is $249. We have an offer on Nutrient Core at the moment because it's new. So just basically I would sign up to Regenerus and see what their price list is for you as a practitioner. In terms of results, it's about 3 weeks from when you send your sample back. It can sometimes be a little bit quicker and sometimes a little bit slower. So that's the tests. And then vaccines. That's such a hard one, and it's such it's such an emotive one as well with vaccines. So detoxification is absolutely a factor in that. But if we knew of a test that could just on its own tell you whether your child would be able to tolerate vaccines, then it would be amazing. But I don't think it's as simple as that. So I think both methylation and detoxification can help with that, but it's a much bigger, more complex picture. And you could see, for example, whether siblings have experienced problems. But I wouldn't rely just on genetic testing for that. And then finally, the estrogen metabolism. So fast, slow metabolism, really the way to look at that is you can look at the genetics and you can see whether certain genetic pathways are likely to be upregulated. But then you could also look at measures and biomarkers of estrogen levels, both the pre-methylated forms of estrogen and the methylated forms of estrogen. So one of the things that Dutch looks at is a measure of 2-hydroxy-estrogen, which is the pre-methylated, and then it also looks at the methylated 2-hydroxy-estrogen, which is after the COMT has acted on it, which gives you an idea of that part of the pathway and how efficient that is. And then there are other things that are further down the pathway, like glucuronidation and sulfonation are both important. But really, high estrogen is a symptom of slow estrogen metabolism. So look at the symptoms on our sample reports. Actually, we do have sample reports available. I think they're going to be on the Regenerus website as well, but they're on Lifecode Gx resources page. And you can see on their sample reports, which include information about symptoms, and aspects of why you might be looking at genetic testing if your client is experiencing certain symptoms, and how to put that together with the genetics. So I think that's it from me. And I don't know about anything else in terms of that's the questions answered. I don't know if everyone can see the panel or the chat, but basically there is going to be a recording, and Regenerous will send out a link to the recording to all the delegates. So thank you very much, and thank you very much to Regenerous for hosting this, and please do get in touch, and I hope to work with you in the future. That's it. Goodbye. And it's 2:10, so thank you for your time.